Interphase. At this point, the cell has a lot of metabolic activity occurring as it prepares for the rest of mitosis. It is during this phase that the cell grows, storing enough of the energy required to fuel cell division. The DNA has now replicated, but not formed a chromosome. It is called chromatin. Prophase. Chromatin, the building blocks of chromosomes, condense. The centrioles of microtubule organisms begin to separate and move toward opposite ends of the cell. The DNA strands shorten and condense to form the chromosome. Metaphase. The membrane of the cells dissolve as the proteins of the cells begin forming kinetochores, and the chromosomes begin moving. The fibers begin to align all of the genetic material, or chromosomes, along the middle of the cell. The genetic material on each side of the cell now should be identical, with each side receiving one copy of each chromosome. Anaphase. The identical chromosomes separate all at the kinetochores formed by the proteins and begin moving to opposite sides of the cell. The fibers shorten as the division of the cell begins. Telophase. The fibers disperse and seem to pull the two cells apart. These fibers then disintegrate. Cytokinesis. The fiber ring made of actin forms around the center of the cell, which completes the process of one cell becoming two. Each new cell has its own nucleus. Interphase. Before a cell can enter cell division, it needs to prepare itself by replicating its genetic information in all of the organelles. All of the preparations are done during interphase. Interphase proceeds in 3 stages: G1, S, and G2. G1. After mitosis is complete, the new daughter cell begins to accelerate its biochemical processes, which were slowed down by mitosis. S. The S phase begins with the replication of cellular DNA, leaving the cell with twice as many chromosomes. G2. During this phase, proteins which are necessary for cell division are synthesized. The chromosomes begin to condense, and the proteins necessary for construction of the mitotic spindle are also synthesized. Prophase 1. Unlike prophase of mitosis, the 2 chromosomes combine or synapse to form tetrads. At this point, the chromosomes cross over at points called chiasmata. Crossing over allows the chromosomes to exchange genetic material, allowing for more different combinations. As in the prophase of mitosis, the nuclear envelope disperses, the spindle moves to the center, and the tetrads become connected to the spindle fibers by kinetochores. Metaphase 1. In metaphase 1, the tetrads are again arranged across the center by the movements of kinetochores, with the 2 centromeres opposite each other, but this time the sister chromatids will not be pulled apart, as in mitosis. Anaphase 1. In anaphase 1, the chromatids holding the chromosomes together loosen. The 2 homologous chromatids on each tetrad are separated into separate poles. Since the chromosomes for each parent can go into either pole, this is another means to increase genetic diversity. Telophase 1. In this phase, like in mitosis, the chromosomes are moved into opposite poles and the nuclear envelope reforms and the spindle is broken down. Remember that there are 2 chromosomes, not 1 as in mitosis. Prophase 2. In meiosis, the cell goes directly from telophase 1 to prophase 2 without the interphase. In prophase 2, the nuclear envelope is again dissolved and the spindle is set up again. Prophase 2 is identical to the prophase of mitosis, except that there is half the amount of chromosomes. Metaphase 2. Again, the chromosomes move into the center and line up. Now there are 2 chromosomes instead of 2 tetrads, so that the chromatids will split off this time. Anaphase 2. The kinetochores move toward the poles, splitting up the sister chromatids. Telophase 2. In telophase 2, the chromatids concentrate in the poles and the nuclear envelope is reformed and the spindle again is dissolved. The cells divide for the last time, leaving a total of 4 cells.